Hello, thank you all for coming to my presentation. I am excited to share with you all what I have been up to this past year, studying how neuromedian U regulates osteoblast differentiation and activity. Here is an overview of this presentation, beginning with a brief introduction on osteoporosis and neuromedian U, followed by what we did to obtain the results, concluding with where I believe the research is headed in the future. Osteoporosis is a multifactorial disease characterized by low bone mineral density, compromised bone strength, and increased risk to bone fractures. When bone resorption is greater than bone formation, bone loss occurs, and over time, this imbalance leads to decreased bone mass. Millions of people worldwide are affected by osteoporosis, and this number will only continue to increase as the world population ages. Osteoporosis not only impedes patients' lives physically, but also burdens them financially. There are many direct and indirect costs associated with osteoporosis, including but not limited to large hospital, medical, and surgical bills, long-term nursing care expenses, and lost workdays. According to the National Osteoporosis Foundation, osteoporosis-related costs is around $19 billion annually and is expected to increase to over $25 billion annually by 2025. Moreover, there is a negative impact on the mental health of patients with osteoporosis. Fractures in older adults can often lead to the need of long-term nursing care, which results in loss of independence, and this can exacerbate feelings of isolation and depression. There is no one-for-all treatment for osteoporosis. Treatment plans often include lifestyle changes that may involve getting enough dietary calcium, taking vitamin D, exercising regularly, fall-proofing the home, and avoid smoking and excessive alcohol. There are several osteoporosis treatment options currently available, and the number of treatments continue to grow. Out of the current pharmacological treatments, bisphosphonates are the most commonly used. Though there are many effective treatment options available, patient adherence is still poor due to concerns over rare side effects and the limited clear evidence supporting long-term efficacy. Therefore, there is still a need to investigate different targets to develop new drugs without these rare side effects and have evidence of prolonged positive effects to bone health. One possible area of research for new targets for osteoporosis therapies is neuromedian U. Neuromedian U is an abundant neuropeptide found in high concentrations in the GI tract and the central nervous system. NMU is involved in several functions in the body, including smooth muscle contraction, blood pressure and blood flow, stress response, and bone remodeling. NMU is highly conserved with many isoforms, and NMU8 and NMU25 are the most common isoforms. NMU also has two receptors, NMU receptor 1 and NMU receptor 2, both of which are G protein coupled receptors that, upon binding, increase intracellular calcium via phospholipase C activation, inhibit forced colon-mediated CAMP accumulation, and activate extracellular signal-related kinase or ERK pathway. Previous studies illustrate NMUR1 deficient mice show no significant difference in bone mass compared to control, suggesting NMU signals through NMUR2. Studies also show NMU directly stimulates proliferation of rat calvarial osteoblast-like cultured cells, likely signaling through NMUR2. Further research on NMU and its receptors is needed to understand how NMU exerts its effects on bone remodeling. For this study, we have 12-week-old female NMU mutant and control mice that were gifted to us by David Artis at Biomedical College, Cornell University. Micro-CT on tibias were performed using high-resolution micro-CT 80 scanner. Bone volume fraction was measured for the tibia mid-diaphyseal region for cortical bone and proximal metaphyseal region for trabecular bone. The cells we chose for this study were Marian bone marrow stromal cells, or the W2017 cells, and Marian calvaria osteoblast-like cells, or the MC3T3E1 cells. To assess how these cells behave and differentiate, we performed alkaline phosphatase activity and proliferation assays and used quantitative RT-PCR to analyze the gene expressions. We also used Phospho Explorer antibody arrays, which allowed us to use small amounts of protein to test for hundreds of downstream targets. To confirm the report of increased trabecular bone mass in NMU mutant mice, micro-CT was performed on 12-week-old female NMU mutant and control mice. Similar to Hsiao et al., we saw an increase in trabecular bone volume fraction in the proximal metaphyseal region of the tibiae of NMU mutant mice compared to wild type. However, no difference was observed in the mid-diaphyseal cortical bone volume fraction of NMU mutant tibiae, shown here in Figure 1a. In Figure 1b, the RT-PCR analysis on RNA from marrow-free humeri of wild-type mice confirmed that NMU and its receptors NMUR1 and NMUR2 is expressed in the postnatal bone microenvironment. This suggests that NMU may regulate bone remodeling directly. To establish an in vitro system to examine the potential for NMU to exert direct effects upon bone cells, we focused on cells of the osteogenic lineage, as Hsiao et al. Reported specific increase in osteoblast activity in global absence of NMU with no change in osteoclastic characteristic parameters. Here in Figure 1c and 1d, RNA obtained from the W2017 and MC3T3E1 cells indicate that these cells express NMU, NMUR1, and NMUR2. We used RT-PCR analysis to see how NMU expression is affected as W2017 cells underwent osteogenesis. Shown here in Figure 2a, W2017 cells have the ability to undergo differentiation, and NMU expression increases as W2017 cells differentiate. This suggests NMU acts as an endogenous negative regulator of osteoblast differentiation. Next, we sought to explore how the inclusion of NMU8 and NMU25 affects W2017 cell differentiation. Here in Figure 2b and 2c, the inclusion of NMU25 throughout differentiation resulted in a decrease in osteoblastic markers, ostrix and osteocalcin, with no difference with NMU8 inclusion. With NMU's ability to repress osteoblastic differentiation of W2017 cells, we hypothesized that NMU would also decrease osteogenesis in MC3T3E1 cells. To test this hypothesis, quantitative RT-PCR were performed on MC3T3E1 cells that underwent osteogenesis in the presence of either NMU8 or NMU25. This resulted in an increase in osteoblastic markers RUNX2 and ostrix, shown here in Figure 3a and 3b. Consistent with the findings of Rzyński et al., NMU pathway activation did not change the expression of osteocalcin. To validate the RT-PCR data, alkaline phosphatase activity and cell proliferation assays were performed. Here in Figure 3c, we see an increase in alkaline phosphatase activity with inclusion of NMU8 or NMU25 during the differentiation process. And here in Figure 3d, like Rzyński et al., we found NMU8 treatment led to an increase in proliferation. Like Hsiao et al., we saw no difference in proliferation with NMU25 treatment. The different effects exerted by NMU8 and NMU25 on osteoblastic marker expression in W2017 cells and MC3T3E1 cells led us to hypothesize that these cell lines have different signaling responses upon NMU pathway activation. Since the molecular mechanisms of the NMU pathway are currently unknown, we took an exploratory approach using multi-analyte phosphoprofiling antibody arrays focusing on those effectors whose phosphorylation status was altered by 50% or more when compared to total levels of said effector. This showed in undifferentiated W2017 cells, NMU25 treatment elicited increased activation-related phosphorylation of many effectors that were mostly unresponsive to NMU8, as shown here in Table 1. However, here in Table 2, we see a very different phosphorylation status with undifferentiated MC3T3E1 cells. There were at least 37 effectors whose phosphorylation status are changed, and 35 of these effectors were not regulated by NMU8 or NMU25 in W2017 cells. For instance, the phosphorylation status of STAT4 was only strongly increased in MC3T3E1 cells and not in the W2017 cells. In fact, activation-related phosphorylation of CD5 and NF-kappa B P65 were oppositely regulated by NMU25 in W2017 and MC3T3E1 cells. The phosphorylation status of CD5 and NF-kappa B are increased in W2017 cells and decreased in MC3T3E1 cells. From this study, the understanding of basic factors controlling osteoblast behavior was advanced. Here we show NMU exerts direct effects on osteoblast progenitor and osteoblast-like cells. Different NMU isoforms seem to have different effects on these cells. NMU25 restricted osteoblast differentiation in W2017 cells. NMU expression in W2017 cells increases with differentiation, and that combined with our earlier data showing global NMU loss in mice display high bone mass together support the idea that NMU is a negative regulator of osteoblast differentiation and raise the possibility that targeting the NMU pathway may be a future therapeutic strategy. These results suggest different NMU isoforms have cell-specific and isoform-specific effects and should be considered when predicting the outcomes of cell-specific knockout in osteoblastic lineage cell lines. Much details on how NMU exerts its effects on osteoblast progenitor and osteoblast-like cells still remains unknown. Particularly, there is not in vivo evidence to support the hypothesis that NMU is a negative regulator of osteoblast differentiation. The results from this study also do not show which of its receptors NMU signals through, or how NMU mounts different signaling patterns in various cell types. Moving forward, I hope to continue to explore NMU and its role in bone remodeling. I propose several next steps, including NMU8 or NMU25 treatment on NMU knockout W2017 cells to support the idea that NMU acts as an endogenous repressor of osteoblastic differentiation. I propose using lentiviral-mediated delivery of short hairpin RNA targeting NMU. A prior study in our lab found that this method is the most effective means of accomplishing knockdown in W2017 cells. Next, I aim to validate the current antibody array data with Western blots. Samples from W2017 and MC3T3E1 cells treated with NMU8 or NMU25 would be assessed by Western blots for targets such as CD5, NF-kappa B P65, and STAT4. Knowing the specific molecular mechanisms of how NMU exerts its effects on these cell lines is one of the first steps in expanding the current understanding of NMU and its role in bone remodeling. NMU treatment on NMUR1 knockdown or NMUR2 knockdown W2017 and MC3T3E1 cells would allow us to determine which receptor NMU primarily uses in the regulation of osteoblastogenesis. Given previous micro-CT analysis of NMUR1 knockout mice do not show any significant difference in bone mass, I anticipate NMU elicits its effects through NMUR2, which would be evidenced by impaired ability of NMUR2 knockdown cells to respond to exogenous NMU treatment. However, it is possible that knockdown of a single receptor results in no defect in NMU responsiveness, suggesting that NMUR1 and NMUR2 may compensate for one another, or that another unknown NMU receptor is involved. Some studies suggest NMU effects on bone are not direct, but rather is a secondary effect from the brain. In contrast, the results from my study show NMU is capable of exerting direct effects on bone cells. To distinguish between these possibilities, I would like to obtain rat bones from where hypothalamic expression of NMU is knocked down. A pilot study of this approach is currently underway in collaboration with Dr. Jonathan Hummel at the University of Texas Medical Branch at Galveston. The bones are currently being analyzed by micro-CT to determine bone mass. I predict that bone mass will be unchanged in hypothalamic NMU knockdown rats, supporting the idea that NMU has a direct effect on bone cells. Future studies involving bone-specific knockdown or knockout of NMU expression would directly address this possibility, but those answers are currently available. Thank you for listening to my presentation, and I would be happy to answer any questions.